Hello students. So we will continue with NCERT chapter 10, which is Biomolecules. And today is theory part 5, and we are going to see NCERT section 10.1.5. This is disclaimer. So first let us see the objective of today's lecture. So after attending this lecture, you will be able to explain the characteristics of biomolecules like carbohydrates, proteins, and nucleic acids and hormones. And you will be able to classify carbohydrates, proteins, nucleic acid, and vitamins on the basis of their structures. Now, according to NCERT, the topics which we will cover today is 10.1.5 Importance of Carbohydrates. Then we will solve in-text question 10.1, 10.2 and 10.3. And we are going to solve NCERT exemplar questions 10 and 16. And at last we will summarize what we have studied in today's lecture. Okay, so let's start with importance of carbohydrates. The first question is, what is the need of carbohydrates? Why do we need carbohydrates? So first see that carbohydrates, they are vital for both plants and animal life. This means that without carbohydrates, living organisms would not be able to survive or function properly. So they are vital. Next, carbohydrates, they make up major component of the human diet. So most of the energy we get from food comes from carbohydrates like rice, bread and potatoes. Now if we talk about the traditional perspective, in Ayurvedic medicine, honey is used as a quick energy source by Veds or Ayurvedic practitioners. Now we are going to discuss some major importance of carbohydrates. First, carbohydrates, they serve as an energy source. So when we eat foods which is rich in carbohydrates, our bodies break them down to release energy. Second, carbohydrates, they are used for energy storage. In animals, excess glucose, it is stored as glycogen in the liver and muscles. While if we talk about plants, so in plants, it is stored as starch. So this stored energy can be released when the body needs it, such as during exercise or fasting. Third is that carbohydrates provide structural material. So in plants, cellulose is a carbohydrate that form the cell wall, giving plants their shape and strength. Fourth, if I talk, then carbohydrates, they are important as industrial raw materials. So they are used to make products like paper, textiles, and even biofuels. Finally, carbohydrates, they are a component of biomolecules. Okay, so these were the some major importance of carbohydrates. Now we'll see the need of carbohydrates. First, we are going to talk about energy source. So carbohydrates make up the majority of the food we eat daily. For example, we eat roti, rice, bread, potatoes. So all are source of carbohydrates. Therefore, we can say that carbohydrates, they play a vital role in our daily diet. So first, if we talk about how significant carbohydrates are, so carbohydrates, they make up a major portion of food which we eat every day. Now, what happens to these carbohydrates in our bodies? So these are broken down into a simpler sugar which is called glucose. And what does this glucose do? This glucose, it acts as fuel for our cells, especially in the brain, muscles, and other organs. Okay, so they are broken down into glucose and they fuel cells. Now it is important to note that carbohydrates, they are essential for optimal brain function as well as for the concentration. Now another thing is that carbohydrates prevent proteins from being used as an energy source. So this means that proteins can focus on their main job. What is their main job? Their main job is building and repairing tissues. So carbohydrates handle the energy needs. So this separation of role is very crucial for maintaining a healthy body. Now if we talk about starch, which is a type of carbohydrate, it is found in cereals like wheat, rice and corn. So starch is a major food supply for people all over the world, providing a steady and reliable source of energy. So if you look at this diagram, you will see that how carbohydrates, they serve as an energy source for humans. Okay, so see, this is the carbohydrates or the food which we eat, and our body breaks down that carbohydrate into glucose, which provides us energy to do a daily work. Okay, so this is the carbohydrates as the energy source for human. Now second, if we talk about energy storage, then whatever we eat daily, the largest portion consists of carbohydrates. Okay, so now we will talk about the energy storage. So the energy storage in animals, so glycogen is the main storage here. If we talk in case of animals, so it's like a reserve tank for energy. And if we talk about plants, then in plants, starch act as the storage molecule. Okay, so you need to remember, in animals, glycogen act as the storage molecules, and in plant, starch act as the storage molecules. Now, if we move on to the structural material, then cellulose is a key structural biomolecule in plants. It forms the cell walls, giving plants their shape and strength. So without cellulose, plants wouldn't be able to stand upright or protect their cells. Next, if we talk about wood, so wood is another example of cellulose. So this cellulose in wood, or I can say the cellulose in wood, it makes it strong and sturdy, which is why we use wood to make furniture and buildings. So this shows that how plant biomolecules, they can be useful even outside the plant. If we talk about cotton fibers, they are also made up of cellulose. Okay, so cotton fiber is cellulosic. So this means that many everyday materials like cotton clothes, they come from plant biomolecules. Now if we talk about industrial raw material, then if we see some specific examples, then biomolecules, they are used to make product like paper, textile, lacquers and even in breweries. Okay, so if you see this, this is used for making all of these things. Okay, next if we talk about the component of biomolecules, then specially we talk about 2 aldopentoses, D-ribose and 2 oxy-D-ribose. So they are found in nucleic acids. Okay, so nucleic acids can be like DNA and RNA. So if you see, this is the structure of a ribose and this is the structure of 2-deoxyribose. Okay, so these are the components of biomolecules which are present in nucleic acids. Okay, now let us solve the NCERT in-text question 10.1. What is the question asking? Glucose or sucrose, they are soluble in water, but cyclohexane or benzene, which is simple six-membered ring compounds, they are insoluble in water. So you have to give the reason. Can you guess what is the reason for this? Okay, so first let's talk about glucose and sucrose. So glucose molecular formula C6H12O6 and sucrose C12H22O11. So both of the glucose and sucrose, they are examples of biomolecules that dissolve easily in water. Okay, so let us see the reason what makes them so compatible with water. First, if you see, glucose and sucrose both have many hydroxyl groups. That is the OH groups. And hydroxyl groups, they are simple oxygen and hydrogen atoms which are bonded together, and they are very good at interacting with the water molecules. Next, if you see, because of these hydroxyl groups, glucose and sucrose, they can form strong hydrogen bonds with water. So if I talk about hydrogen bonding, it is a special interaction between molecules that have hydrogen which is attached to electronegative atoms, okay, like oxygen. So this makes it easy for these sugars to mix and dissolve in water. Now if we talk about benzene, which is C6H6, and cyclohexane, which is C6H12, then they are both six-membered ring compounds. But unlike glucose and sucrose, they do not dissolve in water. So if you see the key difference, it is that benzene and cyclohexane do not have any hydroxyl group. Now, if they don't have any hydroxyl group, they can't interact with water the same way as glucose and sucrose were interacting, okay? Because they don't have any hydroxyl group. So, there is no possibility of hydrogen bonding in case of benzene and cyclohexane. Also, if benzene and cyclohexane, they are non-polar molecules. If we talk about water, water is a polar solvent. So, if benzene and cyclohexane, they are non-polar solute, they won't dissolve in a polar solvent. Because you need to remember a rule that like dissolves like. So, non-polar solute tends to dissolve in non-polar solvent. So, therefore, to sum up, we can say benzene and cyclohexane, they are non-polar in nature, which is why they don't mix with water. So, the presence or the absence of polar group like OH, it makes the difference in the solubility. Now, let us move to the next question. What are the hydrolysis product of A, sucrose, and B, lactose? So, we have to see their hydrolysis product. First, if I talk about sucrose, so when sucrose undergoes hydrolysis, it splits into two simple sugars. It is D-glucose and D-fructose. Okay, so it will give one molecule of D-glucose and another molecule of D-fructose. So if you will see, this is a molecule of sucrose, okay, on hydrolysis in the presence of an enzyme in what is or an acid. This sucrose, you will see, it will react with water and it will produce one molecule of D-glucose and another molecule of D-fructose. Okay, so this is a glucose and this is a fructose. So this is their hydrolysis product. Now if we talk about lactose, then lactose is hydrolyzed. It splits into two simpler sugars. One is beta-D-galactose and another one is beta-D-glucose. So if you look at this reaction, this is a lactose. In the presence of water and the acid, you can see in the presence of enzyme lactase, then you will see it splits into beta-D-galactose and beta-D-glucose. So if you see, each product has same formula, but it has a different structure. Okay, So it will split into beta D galactose and beta D glucose. Now moving on, how do you explain the absence of aldehyde group in the pentaacetate of D-glucose? Okay, so you have to explain the absence of aldehyde group. Now if you notice that glucose doesn't usually exist as a chain molecule in solution. Instead, it forms a ring structure which is called as cyclic hemiacetal. This means that the aldehyde group at the end interacts with an alcohol group on the same molecule, creating a ring, and it basically hides the aldehyde group. Okay, so you can see it predominantly exists at cyclic hemiacetal form in the solution. So if you see the conversion of glucose to pentaacetate, this is glucose, okay? And when you treat it with acetic anhydride, it forms glucose pentaacetate. Okay, so at carbon 1, you are seeing you have an OAC group which is present over here. So if you see about D-glucose pentaacetate, then what happens? During the reaction, all the 5 hydroxyl groups on glucose. So if you see all the 5 hydroxyl groups which is present on glucose, they are replaced by these OAC groups, okay, or by the acetyl groups. So this means that OH, it gets converted to an O C double bond O CH3 group, okay. So this replacement is crucial for understanding that why aldehyde groups can't be present in the final product. So if you will see over here, all the 5 hydroxyl group, they are replaced by the acetyl groups. Okay. So the conversion of all the hydroxyl groups to the acetyl groups, it locks the glucose molecule in its ring form. And this prevents the ring from opening up to reveal the aldehyde group. Okay, which is why pentaacetate doesn't show any aldehyde properties. Okay. Now, if we talk about the glucose, if you notice in glucose pentaacetate, there is no hydroxyl group at carbon 1 position. Okay, so if you'll see in the case of glucose pentaacetate, there is no free OH group at carbon 1, which is also called as the anomeric carbon. Okay, so this shows you that all the hydroxyl group, including the C1 carbon, has been converted into acetate groups. So there is no free OH group that is present. Now if we focus on the anomeric carbon or C1 carbon, in the original glucose molecule, this carbon has 5 hydroxyl group. So if you look at this original glucose molecule, these have 5 free hydroxyl group. But in the case of pentaacetate, even this group is also acetylated. That is the C1 carbon. Okay, this OH which is present at C1 carbon, this is also acetylated. This means it is being replaced by an acetyl group. So it is no longer available to participate in reactions that require a free OH group. Okay, so you will see there is no free hydroxyl group at carbon 1, as free hydroxyl group at carbon 1 has also been acetylated. So you will see because the hydroxyl group at C1 is no longer free, the molecule can't switch back to its open chain form. Okay, and remember one thing, that open chain form is necessary for the aldehyde group to be exposed and react. Without this ability, the aldehyde group stays hidden. Okay, so now the molecule cannot revert to the open chain form. And as a result, glucose pentaacetate doesn't give a positive result in Felling's or Tollens test. Okay, and these tests, they are used to detect the presence of free aldehyde group. So if the molecule can't revert to its open chain form, it won't react in these tests. Okay, that is the Felling's test and the Tollens test. So what we can conclude? To sum up, we can say that the reactive aldehyde group is absent in glucose pentaacetate. This is because all the hydroxyl groups, including the one at the anomeric carbon, are acetylated, which prevents the molecule from showing a typical aldehyde reactions. Okay? Now let us move to the next question. Which of the following statement is not true about glucose? So you have been given 4 options and you have to tell which is not true. First, it is an aldohexose. B, on heating with HI, it forms N-hexane. C, it is present in furanose form. And D, it does not give 2,4 DNP test. So tell me the answer that which is not true about glucose. Okay, if we see the correct answer, then statement C, it is saying it is present in furanose form. Okay, one by one, let's discuss. First, we will analyze all the options, all the 4 options, one by one. First, let us move to first option. It is saying it is an aldohexose. So what do you think? Is this statement true or wrong? This statement is absolutely true. Okay, glucose is an aldohexose because it has 6 carbon atoms. Hex, what does hex mean? Hex means 6. So glucose has 6 carbon atoms and it contains an aldehyde group, which is a specific arrangement of atoms at one end of the molecule. So the option A is a true statement about glucose. Okay? And if you want to remember, it is very simple that aldo refers to aldehyde group. If you look aldohexose, it is very simple. Aldo means aldehyde group and hexose refers to 6 carbon. So from the name itself, you can justify that it contains an aldehyde group and it has 6 carbon atoms. Okay, so glucose is a aldohexose. So if you see, it has 6 carbon atoms, 1, 2, 3, 4, 5, 6. And it has this aldehyde group. Okay. Now if we see option number B, on heating with HI, it forms N-hexane. So this statement is also absolutely true. When glucose is heated with HI or hydrogen iodide, it produces N-hexane. So if you see, this is glucose. When you are heating with HI, it gives N-hexane. Okay. So this transformation shows that glucose molecule is converted into N-hexane. Okay, so if you see this reaction, it removes all the oxygen atoms from the glucose, leaving behind a simple hydrocarbon chain. So this is formation of N-hexane. Now if we move on to the next option, which is option number C, it is present in furanose form. Now this statement is wrong. Okay, glucose, if we see, glucose is actually present in pyranose form and not the furanose form. If we notice the structure, okay, this is a 6-membered ring and this is a characteristic of pyranose. So this is important because the shape of the molecule affects how it interacts with the other molecules. Okay, so this is present in pyranose form, not in the furanose form. So if I talk about pyranose refers to a 6-membered ring structure that includes 1 oxygen atom. Okay, and this is very similar to the chemical compound pyran. And this is different from furanose. Furanose is actually a 5-membered ring. But if I talk about pyranose, this is a 6-membered ring. And this has 1 oxygen atom. Okay. Now if I talk about D option, it is saying it does not give 2,4 DNP test. So the 2,4 DNP test, this is used to detect the presence of an aldehyde or ketone group in the molecule. So if you see, if you're talking about the cyclic structure, then glucose in its cyclic structure does not have a free aldehyde group which is available to react in 2,4 DNP test. Look at this cyclic form. There is no free aldehyde group which is present. Okay. So if you'll see, this is the ring form of glucose where the aldehyde group is involved in the formation of the ring. So it can't react as a free group. Okay. So this explains that why glucose doesn't give a positive result in this test. Okay, so therefore, if we sum up, then option number C is the right answer for this question. Okay, now let us move on to the next question. Which of the following reactions of glucose can be explained only by its cyclic structure? First, glucose forms pentacetate. Glucose reacts with hydroxylamine to form an oxime. Pentacetate of glucose does not react with hydroxylamine. And last one, glucose is oxidized by bromine water to give gluconic acid. So you have to tell which can be explained only by its cyclic structure. Okay, so can you guess what is the right answer? Which of them can be explained only by its cyclic structure? So if you see, the right answer for this question is C. Pentacetate of glucose does not react with hydroxylamine. So one by one, let's discuss. First, A option, glucose forms pentacetate. So there are 5 hydroxyl groups which is present in the open chain form of glucose. So when glucose reacts with acetic anhydride, all the 5 hydroxyl groups can react to form a compound called pentacetate. Okay, so if you will see, there are 4 here and 1 here. There are 5 free OH groups that are present. And when this reacts with acetic anhydride, all the 5 OH groups, they react to form a compound which is called glucose pentacetate. So this is glucose pentacetate. Okay, so you will see all the 5 OH groups has been replaced by acetate. Okay, so this process is possible because all the 5 OH groups, they are accessible in the open chain form of glucose. So this can be explained by the open structure of glucose. Okay, so therefore this option is correct option. Next, if we see the B1, it is saying glucose reacts with hydroxylamine to form an oxime. So in this, we will see that the aldehyde group, which is the CHO, it is present in the open chain form of glucose. So the aldehyde group is a carbon atom which is double bonded to oxygen and it is single bonded to hydrogen. Okay, so this group is reactive. This is responsible for certain chemical reactions. So this aldehyde group is in the open chain form of glucose. So when glucose reacts with hydroxylamine, which is NH2OH, the aldehyde group forms a compound which is called oxime. Look here, this aldehyde group forms a compound which is called an oxime. Okay, so this shows that the open form of glucose is necessary for this reaction to happen. Okay, so this can be explained by the open structure of glucose and not the cyclic form. The presence of aldehyde group in the open chain structure is what allows glucose to react with hydroxylamine and form an oxime. Okay. Now if we talk about option C, that is the pentacetate of glucose does not react with hydroxylamine. So if you notice here, only the cyclic structure of glucose explains the absence of free aldehyde group. In the open chain, this group is present and it would react, but in cyclic form, it is not available for reaction with hydroxylamine. Okay, so you will see this cannot be explained by the open structure of glucose. Okay, so this means that the lack of the reaction cannot be explained by open structure of glucose. So if you see the image over here, you will see that the CHO group, which is the aldehyde group, is no longer free. Okay, so this is why that this reaction can only be explained in its cyclic structure. Okay. If I talk about option number D, then glucose is oxidized by bromine water to give gluconic acid. So here the oxidation specifically targets the aldehyde group, okay, in the open chain form of glucose. So this group is reactive and it is responsible for chemical properties. Okay, so you will see when it is exposed with bromine water, this group is converted. So if you will see the aldehyde group over here, it is being oxidized to carboxylic group. So this forms a gluconic acid. Okay, so this can be explained by the open chain structure of glucose and not the cyclic form. Okay, so the aldehyde group is only available for oxidation when glucose is in its open form. That is why this reaction does not require the cyclic structure. Okay, so therefore option number C is the correct answer. Okay, if you'll see, only certain reactions of glucose like mutarotation and formation of glycosides, they can be explained by cyclic structures. Okay, now let's summarize what we have studied in today's lecture. First, we talk about carbohydrates. So they are of immense importance in everyday life as well as in nature. Okay. Next, carbohydrates, they are very much vital. First, they act as a main source of energy. So they fuel our cells, especially the brain, which relies almost entirely on glucose. Next, if we talk about storage, in animals, carbohydrates are stored as glycogen, which is mainly in liver and muscles, and as starch in plant. Next, if we talk about structure, so in plants, the cellulose is a carbohydrate that makes up the cell walls, giving plants their shape and strength. Okay, and if we talk about wood and cotton, they are mostly cellulose. Next, they are carbohydrates. They are important in industry also. So they are used to make paper, textiles, and even lacquers. So when you are writing on a paper or you wear cotton clothes, you are using the products which is made of carbohydrates. And next, finally, carbohydrates, they are key components of biomolecules like DNA and RNA. So thank you so much, students, for your attention. I hope this lesson was helpful to you. Thank you.